morning, everyone. We're ready to start our next session. My name is Kristen Chisholm, and I am a very proud member of the Yerbury Lab Group here at Wollongong University, and I'm honoured to be introducing our next two speakers this morning. Our first speaker has been a mate of mine since we were 12 years old. She was Justin's wife, partner in family life and global adventures, and ultimately his carer and advocate. Rachel's talk today is titled Lived Experience Insights. And the sad truth is that there is probably no one more perfectly equipped to give us this information today, because there wouldn't be too many people with Rachel's experience and who knows the intimate and heartbreaking journey this disease takes patients and their families on. But what is equally true is that without Rachel's personal strength, her courage, and her support of Justin, we would not have his brilliant contributions to this field, bringing hope to other families suffering from these diseases. I'm so proud to be part of this family's legacy, and I'm proud to call Rach my friend.
Thanks, Chris. I really appreciate that intro. And I've also been very grateful to have you by my side throughout this journey. So I'd like to start by acknowledging the first Australians whose lands we live and work on. I'm zooming in today from Yorta Yorta lands in the Goulburn Murray region, and I'd like to pay my respects to elders past, present and emerging for their care of these lands, waters and wildlife, including the mighty Murray River system and their totem, the long-necked turtle. You would have experienced the welcome to country yesterday by my friend Jodie Edwards, but I'd also like to honour myself, the Wadi Wadi, Dharawal and Alauri people of the Dharawal Nation, where the conference is being held and where Justin and I live most of our lives. I'd also like to thank the organisers of this conference for dedicating this morning's session to honour my much-loved husband, Justin, and for asking me to share my lived experiences of this horrendous disease. I also want to thank everyone who is attending this session today, those in person at Wollongong and those online, the researchers, academics, caregivers, and most of all, those of you who are living with this disease. Today, I will share my lived experiences of MND. In doing so, I hope to acknowledge and give voice to those of you who, like our family, know firsthand the devastation this disease wreaks. I hope to offer a context and a meaning to the work of you tireless researchers who dedicate yourselves to this cause. As I go along, I will offer some learnings I have gained throughout this journey. I hope that they may be relevant to both researchers and to community members. For many of you, my words will be hard to hear. If it becomes too confronting, please feel free to leave the room or do what you need to do to be okay. My involvement in MND came from sharing a life with Justin, the love of my life. The main experience was helping him to navigate his own disease between 2016 and 2023. But there were other experiences too, like watching in fear as MND ravaged the family, like supporting his passion and drive to become Wollongong's first MND researcher, and then later advocating and fighting for his rights in society, with government, and in academia. But what came first was the family story. the pain and suffering of which continue for those of us who are left bereaved and traumatized by this terrible disease. When I reflect on the family, I think about Justin's little sister, Sarah, who died from MND when she was 26, and Justin's mum, who died aged 52. His nan and Auntie Kathy, who died within 6 weeks of his mum. Justin's cousin, Ashley, Kathy's son, who was barely into his adulthood when he died 3 months after diagnosis. Ashley's sisters, Stacey in her 20s and Hayley, a new mum in her 30s, who lived with MND at the same time that Justin did. I'm also reflecting about Uncle Ken, who was the first in over 20 years to be diagnosed in 1994. The one who had his arm in a sling at our wedding and died a few months later. MND has taken all but one of Justin's mother's siblings, many of his cousins, several aunts, second cousins, and the list goes on. How much loss can one family take? Not all people living with MND have a familial link. And as you know, the SOD1 mutation is particularly devastating. But even one person in a family is one too many to lose. The first lesson I learned through this MND journey was how to combat my daily fears of this disease. And for me, that was to take one day at a time and to live life to the fullest. Once we realised that Justin had the gene, the first thing we did was go out and buy a canoe. Now that may sound random, but that red canoe was the start of our decision to enjoy every minute of our family time together. And we did. We had parties, camping trips, adventures, overseas holidays. And not only did we do it, but we gave thanks. We were so grateful for our lives. Meanwhile, Justin was also driven to contribute to the field in his hope to find a cure for the disease that afflicted his family. He did a science degree, then a PhD to begin researching MND. He quickly became successful in proteostasis research, and he was the beacon of hope for the family. This pressure in itself would be unbearable for most people, but Justin felt privileged to be able to commit his life to research. And that leads me to the second lesson of this journey, and that is, commit yourself to a meaningful life. For Justin and I, this was predominantly precious time with our family. We found meaning in other pursuits too. For Justin, that was his research. For me, it was embarking on a PhD that I'd always wanted to do. And more recently, for both of us, it was writing our memoirs. When Justin was diagnosed with MND in the week of his 42nd birthday, I was overcome with hopelessness and despair. My cries echoed down the hall of Macquarie Neurology as Dr. Dom Rowe and Justin tried to polish the turd of the terminal diagnosis. They told me it would be okay, but I knew it wouldn't. I had seen too many times how this played out. My world was crumbling around me. Justin, however, still managed to find positives, gratefulness, and hope. He kept working to find a cure with even more energy and passion than he had before. His drive and hope kept us all afloat. Which leads to lesson 3 on this MND journey. Do not underestimate the power of hope and positivity. Without it, MND will drown you, as I have seen it do to too many people. This applies to all of us, no matter how we are involved in this space. There are so many different presentations of MND that it almost seems to be a different disease. Stephen Hawking lived for 50 years with MND. In our family, the age of diagnosis, location of onset, and length of life has varied significantly. From Ashley, who experienced vulvar onset and died within 3 months, to Auntie Bub and Auntie Suzanne, who lived for more than 10 years after diagnosis. Justin's journey was a bit different to others because after meeting Stephen Hawking in Cambridge in 2016 and seeing how he lived on a ventilator, Justin also decided to have life-saving laryngectomy surgery. This was necessary because his respiratory distress had become acute just a year and a half after his diagnosis. Justin's deterioration had been quite slow at first, but in the second half of 2017, the disease started to ravage his body. My life narrowed to solely focus on his medical, health, and increasing mobility needs. He required help to do absolutely everything. Every week, and often every day, Justin would lose another significant aspect of his functioning, often without forewarning. I was always on my toes, problem solving the constantly changing needs, altering routines, support, furniture, equipment. It was a full-time job just to keep his life together, let alone to look after him and keep him healthy. I finally realized that I needed help and support to do this job. And this is lesson 4. Don't try to do this on your own. Whether you are a carer, a person living with MND, or a researcher, you need a team around you. Surround yourself with people who can be there for you, to support, to encourage, to affirm. Find your village, those who have your back. And most importantly, those who can pick you up when you fall, because you will. This disease compromises even the strongest of us. Justin never wanted to be a burden, and he also didn't want to leave us. So he lived and worked for as long as he could. After the laryngectomy and mandatory ventilation in January 2018, he was in ICU for 6 months. He fought infection after infection and struggled to heal. But it wasn't only his health that was a struggle. We had to fight for funding from both New South Wales Health and from the NDIS, and then fight to find suitable carers. Once home, Justin had around-the-clock care and this meant our family had to get used to having people in our homes 24/7, often several people at once. This was extremely hard for me, as I felt uncomfortable in my home most of the time. The daily business of a hospital-like house, constantly fighting the NDIS, managing Justin's life, and providing emotional support was undeniably a huge toll on us both. We needed to be very resilient to cope with this life. So the fifth lesson is gather your internal strength by finding what it is in your life that keeps you buoyant. MND is a hard journey, and you need to look after yourself so that you have enough fortitude to weather the storm. What we learned on our journey is that we had to be able to constantly fight to have the surgery, fight to get funding, fight to have suitable carers, and fight against discrimination. So remember to care for you. Get enough sleep, eat well, meditate, learn to relax and connect with yourself, with loved ones, and with whatever or whoever nourishes you. For me, that's connecting with nature, especially the ocean. Spending time immersed under the water and watching the sea life gives me much needed respite and comfort during tough times. Despite the challenges of the past 7 years, it was also a privilege to support Justin's prolonged life. Justin returned to uni and his research, managing his ever-growing research team and writing grants and papers with his eye gaze software. He had 5.5 bonus years of life in which he watched our girls grow up and spent valuable family time. As you know, despite being paralysed and ventilated, he not only furthered his research, but he also had monumental achievements. He was awarded a member of the Order of Australia. He received the Eureka Prize, the New South Wales Premier's Science Prize, the Keys to the City of Wollongong, and Citizen of the Year in Wollongong. He fought against discrimination and ableism in many facets of society. So everything that we experienced and the burdens I helped Justin bear were worth it. But as a carer or even a researcher, MND cannot be your whole life. And this brings me to the sixth lesson. You need a life outside of MND. This is something that Justin and I always knew was important. We ensured that MND didn't define who we were by having a life outside of the disease. Whether you are a researcher or have lived experience, find your identity as a person apart from MND. MND has a habit of consuming us, taking over our lives and sapping all of our energy. It is easy to lose yourself. So find ways that you can be you. Do what you did before and enjoy what you used to in your pre-MND world. Give yourself permission to enjoy your life. Allow yourself space to breathe, to listen to the birds, to watch the waves. to smell the flowers. The final lesson is to remind yourself that you are doing the best that you can. It is a tough gig living and working in the MND space, and it may feel like you're taking 2 steps back for every step forward. Hang in there. I won't tell you it will be easy. It simply will not. But trust yourself that decisions that you are making today are the right ones for what is on your plate right now. So in summary, find meaning and comfort in your life. Cherish each day. Surround yourselves with love and support. Keep hopeful and be kind to yourself. Know that you are doing the best that you can. Thank you all for listening to me today. To those with lived experience, I hope my talk has helped you to feel validated and that you are not alone. To the researchers who are studying MND and have been for some time, thank you for not giving up, despite the slow grind of research and funding. I want to especially acknowledge those researchers who are continuing to carry the torch and the flame that Justin began at Wollongong. Thank you. I think I can speak for all of us with lived experience when I say that your work means so much and allows us all a much needed glimmer of hope. Thank you.
So I want to thank Rachel for her talk. It would not have been easy. Reliving the journey that she's been on must break her heart every day, and we are grateful for what she offers us. She's available for questions if anyone has any questions that they would like to ask. Might be getting off the hook, Rach. Do we have any online?
I can't see the crowd, so there we go. Hello. It was actually easier to do it to just a blank screen. So
Yes, you did a great job. We're very happy to hear from you.
thanks, Chris. Maybe they're speechless.
Thank you.
That's probably the best description of living with MND as a non-experienced person I've ever heard. So thank you very much. I'm a neurologist looking after people with MND, and I just wanted to tap into your insights, really. You talk about living in the present, which I think is something that many of us advise people to do, and it's something that people with MND tell us all the time is how they cope. As clinicians, unfortunately, we drag people into the future quite regularly in our discussions. You and Justin obviously had to make some very difficult decisions and plan for his medical care and care at home. Do you have any help that you can offer in terms of how people can elide from one of those settings of being in the present into dipping into the future and then still maintaining their peace?
Yeah, thanks for the question. I think it's so important that you're thinking about that. And I guess I think that everyone's different in terms of what they need. So it's really about checking in with the individual and saying, what do you need right now? Dom was certainly the kind of doctor that didn't put us too far into the future. And we were able to sort of take a day at a time, and that really suited Justin. That's his kind of personality. I think some people like myself, probably to know a little bit about the future and what to expect, might have actually been reassuring. So I think it's really about working out where someone's at, where their family's at, and what they need right now, and just to take it slowly and keep checking in.
We did have one online question asking you, Rach, how did you relax? With everything going on, how did you relax?
So yeah, spending time at the beach, in the ocean, snorkeling. I would just, rock up at Port Kembla Beach and put my chair out and just sit there for hours, just watching the waves, swimming. Yeah, that's my thing. I think everyone needs to find their own way. But I do advocate nature as therapy.
We've got a couple more questions, and what's your PhD in, Rach?
So my PhD was around people's connection with nature and how that benefits their psychological well-being, but also about reciprocity and how we can give back to nature.
Hi, Rachel. I'm Rebecca. I'm a speech pathologist from Flinders University in Adelaide. Thank you so much. That was such a beautiful talk to listen to, very moving. My question is, and one of the things that really affected me, I guess, in your talk was about how uncomfortable you felt having a care team within your home. What could make that better for you, or what could make that better for people that are experiencing that?
I guess my short answer is nothing. It's always going to be difficult. I think that people who have separate parts of their house may find it a little bit easier, and certainly I was able to figure out pretty quickly that I needed to have a space where the care team didn't go at all, which included my bedroom and some space out the back. And I think that was really important. We also found it really helpful for Justin to have his own space all in the same room. So we installed, we were fortunate. We were privileged enough to have a huge space for him. So we were able to have a space where we could put a little kitchenette. He had his bathroom, and that sort of limited the amount of care staff that needed to be like in my kitchen and things like that. And that worked really well. And we also we were lucky enough to have an external door where the carers could come in and out of the house. So it was less disruptive. But, you know, I felt uncomfortable, but I also got along really well with all the carers. And I think that that's really an important thing, is that the carers are really aware of exactly what needs to happen in your house, and that as a family member or as a person living with MND, you feel like that you have control over what happens in your house. Because like for me, that was just so important, that sense of that, you know, it was still my house. But, you know, even after all those things were put in place, because we had, you know, more than 5 years of this happening, it was still tough. But yeah, thanks for asking.
One last question from online is, when is your memoir being released?
I don't have a publisher yet. Yeah, so, but you can read Justin's memoir and it's really good.
Thank you, Rach. If anyone has any more questions, if you contact Lizanne, she can pass them on to me and I'll send them on to Rach. But thank you for your time. Go and find some more waterfalls.
Thanks.
So in science, we need brilliant minds like Justin's who can see the needle of relevance in the haystack of biological noise and uncertainty. Our second speaker this morning is also a brilliant science thinker. With a 20-year research career both in Australia and abroad, Darren Saunders has made significant contributions to the field of breast and pancreatic cancer, and since he started collaborating with Justin, the MND field as well. However, science and brilliant thinkers cannot operate alone. Without the ability to communicate the importance of scientific research, the public don't understand, and the policymakers and funders do not prioritise our work. And we end up with the society straying from evidence-based decision-making. In his new role as Deputy Chief Scientist, New South Wales, Darren now walks in the education and communications space. And as the world degenerates into populist political ideas, we need his analytical advocacy more than ever. I'd love to share some interesting and juicy gossip about Darren, but seeing as he is still my PhD supervisor for a few more months, you will need to come back and talk to me next year, and I might tell you a bit more, Darren.
Thanks, Chris. And I'm going to borrow that introduction if that's all right. That was lovely. Thank you. Kristen and I we've known each other since we were undergrads, and we work on a principle of mutually assured destruction. So I know as many secrets about her as she knows about me, and that's the way we we do it. Let me just get this running. Getting old is awful. Okay, I don't know what I did to upset you, Lizanne, but thanks for putting me in a slot after Rick and Rachel. That's not an easy job. Look, one thing I've learnt, but thank you for the invitation. I really do appreciate it. And it is always lovely to come back to Wollongong. It's my second home. And I've learnt over the years when I do talks to do the acknowledgements at the start. Because by the time you get to the end, people are thinking about the next talk or thinking about their questions, and the acknowledgements get lost. So the first acknowledgement I want to make is to Rachel, and I'm going to try and get through this without tears, and I'll do my best. Whole Yerbury family, I'm going to talk about some of Justin's research and reflect on some of the things I learned working with Justin as a scientist. And I know a lot of you in the room have that same experience. Justin did what he did for his family, but he couldn't have done it without his family. They were the absolute bedrock of his work, and they were as much a part of it as any of us as scientists in the room. And Rachel, I think you captured that really beautifully, so thank you. The second acknowledgement I want to make is to the Dharawal people. I grew up on Dharawal land, I studied on Dharawal land. It was a formative experience in becoming a scientist. I spent, like Rachel, a lot of my time in nature. That's why I became a biologist. And more recently, I'm going to talk about this in a minute, I've spent a lot of time out in far western New South Wales in my new role, working on the lands of the Barkindji people, trying to understand why fish keep dying in millions in the Darling Barker River. And it's been an incredible experience to sit and learn from the traditional owners of the land out there, and to understand that they carry with them this incredible scientific knowledge that exists in a form that scientists aren't used to hearing. And it takes a long time to learn how to hear that knowledge and to learn from that knowledge. So I want to acknowledge them for sharing that knowledge in a very gracious way. They've told that story so many times. having a lot of fun and reflecting on challenges. And I found myself one day sitting in the library in a place called Vail in Colorado, which is this beautiful ski resort where movie stars go to ski. And they had this incredible, amazing library next to this little running river with the snow coming down and the pine trees. And I was sitting in a big leather lounge reading Nature in a ski resort, right? What a geek. But what I realized is doing that, my brain was turning to mush, right? My brain was literally turning to mush. And so I thought, okay, time to get serious. Came back to Australia. And I was reflecting on this with Kristen yesterday afternoon. I literally sent 3 emails to 3 heads of institutes in Sydney and said, hi, I'd really like a job as a scientist, please. And one of them gave me a job. And it doesn't work like that anymore, right? So that was pretty lucky. I had a fantastic time at the Garvan Institute working on cancer research for a long time. I then went to Vancouver, lived in Vancouver for 3 years, not coincidentally close to the mountains, and had an amazing time as a postdoc in a lab that had almost unlimited budgets, where Nobel laureates would come wandering through the door on a regular basis and just sit down and have a chat about what you were doing. A really amazing scientific experience. Got homesick, got sick of snowy weather and moved back to Sydney, set up my lab at the Garvan Institute, did that for years, moved across to UNSW, less said about that, the better. And when COVID hit, I got out of there. So I had achieved what a lot of people in science want to do. I got a tenured position. I had a job without an end that paid me to do my job. Now, that's a pretty rare thing in science these days. And about a year after I got it, I walked away from it. We can talk about it, it's a different talk. But what I did was go and set myself up as a consultant. It was a pretty challenging thing to do. It was a lot of fun. I basically started a small business, and you can see on the screen there on the right-hand side, there's all the different companies I worked with. So some of them were biotech companies, they're mining companies. I worked with airlines, police forces, you name it, doing research in a different way. So I had to reframe my skills and experience. So I was presenting research, doing research in a corporate environment, very different environment. And at the same time, doing a lot of communication. So I did a lot of work with the ABC and Channel 7, talking about science. And that became particularly important during COVID, right? A time where science became a spectator sport. Everyone was watching what we all did on a daily basis. And everyone got a very different understanding of how science works, right? It's not that linear path that everyone thinks it is. The way it's presented in movies where you have the eureka idea and then you go and make the drug and everyone gets better. We all know it doesn't work that way. It's a lot of forwards and backwards and a lot of going around in circles and dead ends. And I spent a lot of time helping people understand that. And that's great fun. It's also terrifying. And then earlier this year, a job came up in the office of the chief scientist and engineer. And I'm going to talk a bit more about what that job is, but it really is both the most challenging and exciting job I've ever done. And here's the sort of stuff we do. So I thought, and talking to Lizanne, I thought it's probably a little bit opaque what the office of the chief scientist does. So I thought I'd talk about it for a few minutes. We basically, there's a team of about 35 people up in Sydney, and we are kind of like an internal independent consultant to government. So when any minister in the government has a wicked problem they don't know how to solve, they'll come and knock on the chief scientist's door and say, hey, can you help us fix this problem? I can give you some examples, but the most pertinent example, I started working this job the week before the most recent state election. And the day after the election, the first announcement the Premier made was to get on a plane and go to Menindee, where a huge flood had happened and 20 million fish died in the river, and said, we're going to get the chief scientist to go and figure out what's happened here. Now, I learned about that because I was watching ABC. It's a bit like Utopia in my office, right? We have ABC 24 going in the corner. I'm saying, oh, what's the Premier talking about? And then he said, oh, we're going to get the chief scientist to do a fish review. Okay, guys, we're working on fish. That's like literally how it worked, right? So we do independent advice. We do a lot of independent advice. It's all evidence-based, it's scientific. We don't make policy, we draw a firm line. Other people make policy, we deliver advice based on evidence. That's the line. What the politicians do and the policymakers do with that evidence is a part of the democratic process. It's essentially non-scientific, so we don't cross into there. We do a lot of research support. So we do funding of all sorts of things. We fund conferences, we fund research programs, we build networks. So we develop industries by connecting scientists together with industries and startups and things like that. That's a really important part of what we do. A lot of it is aimed at translation and commercialization, right? So we have to align what we do with political imperatives around building skilled workforces, around advanced manufacturing, making lots of good jobs that pay well for people. And then we do a lot of science outreach. We sponsor a lot of STEM education programs and things like that. And really the whole point is up there in the blue box at the top. It's about using or enabling the development and application of science and engineering and technology. And it's all about improving the economic, environmental and social conditions for the citizens of New South Wales, right? And we take that part very seriously. That's what we do. Okay. It's all driven by a strategy. I'm not going to talk about this in too much detail other than to say it's good to have a strategy. right? So our strategy is based on an evidence-based analysis of what New South Wales is good at, what we have opportunities in, and it really comes down to 4 themes around digital, around energy, around biotechnology, and around materials. My boss, the Chief Scientist and Engineer, is a nuclear physicist by training. So he does all the hard stuff, I do all the squishy stuff. We have a good complementarity of physics and biology, if you like. And the office is full of people with, you know, there's chemistry PhDs, there's civil engineers, chemical engineers, electrical engineers, there's lawyers. It's a really broad mix of people. And then we apply that across all these different industry sectors. And again, I won't bore you with the detail there, but one of the big issues that we've been working on is this thing called the Menindee fish deaths, right? And that picture there, all that white stuff on the top of the river are dead fish. So if you can imagine that river, 40-kilometre stretch of that river, it looked like that. The estimates vary somewhere between 20 and 30 million fish. So on the morning of 17th of March this year, the residents of Menindee woke up to that. It was a river the day before, and then it was a graveyard the next day. In fact, I actually used, and Luke, you'll love this story, I actually used the software that we use to count cells, machine learning algorithms we count cells with. I got hold of the ABC News guys' drone footage of the river, and I fed it through the cell counting algorithms and actually estimated how many fish there were based on that. My boss thought that was a pretty interesting approach. So on the right there are 4 words. When I went into this job, I was very naively thinking, right, we're going to go and figure out the science behind why the fish died. Spending a lot of time in Menindee, we very quickly figured out that it was not just a scientific question, right? It's a social question. The impact of those fish deaths on people's mental health, on their physical health, on the social fabric of the town out there, on their economic well-being of that town is absolutely huge. And so figuring out the science was also about helping the community and helping the people out there. And the other really interesting thing I found is that, I mean, you know, I've got a degree in biology, so I'm not completely unaware of what happens in a river environment. But what actually unlocked this project for me was to use my biochemistry and physiology, because it's essentially a mass balance problem. The fish died because they ran out of oxygen. And so I actually took a biochemical physiological approach to figuring this out. And it looks like this, right? So if you think of the river as a giant fish tank, there are things that put oxygen in and there are things that take oxygen out. And essentially what happened was the demand exceeded the supply, the fish ran out of oxygen and they all starved to death. And it happened very, very quickly. And so you can quantify all of that, you can build models. The other thing we quickly realized is all those dead fish sank to the bottom of the river. They're still sitting in the bottom of the river. They are decomposing and that decomposition uses oxygen. So as soon as the weather warms up, which it's doing right now, we are probably going to have another one of those mass fish kills. So we're in a feed-forward loop, and everyone who's done biochemistry knows what a feed-forward loop looks like. So that's how we got to the problem. And then on top of that, we did some conceptual mapping. So everyone in my office freaked out when I showed them this, but this is the conceptual map we used to try and figure out what levers can we pull to stop this happening? Because one of our terms of reference was, how do we stop this? There's some levers to pull. Most of them, we don't have any data on any of these things, but at least it builds a concept in our minds of where are the things we can do to change things. So the advice we can give to the people who make policy is, how do you fix this? Well, you've got all these levers to pull, start spending money on these things and fix it. And again, using that biochemical kind of analogy, you can look at ways of increasing supply and ways of decreasing demand. The report's public, you can read it. I won't bore you with the details. So the second thing that I've been working on a lot is something that is much closer to the interests of this conference, right? Everyone knows about the amazing breakthroughs of all the new COVID vaccines, largely based on mRNA technology, right? Most jurisdictions in Australia are now have a very strong interest in building sovereign capacity and the ability to manufacture clinical grade mRNA therapeutics and vaccines. Because one thing we figured out in COVID, we have to make it ourselves. It's very hard to get it from overseas. We need to build capacity in doing that. We need the skills to go with that, the supply chains and everything else. So the New South Wales government has put money behind setting up a facility to manufacture clinical grade mRNA and the associated delivery technology, nanoparticles, all that sort of stuff. It's all about not just human health, but animal health. So protecting, you know, really important export and job creating industries like beef and sheep and all that sort of stuff, who get all of these horrible, nasty diseases that will kill our export industries overnight. So it's about building up resilience and biosecurity. It's about building collaboration and research commercialization. And the way we're doing it, the approach we're taking is to build an ecosystem. So the government's building a manufacturing facility. We're setting up a research fund to go with that, and then support for training the people to come through that system, and then building networks of researchers around that. And it's a very interesting business model, which I'll talk about in a sec. So RNA technology has all these amazing, COVID really showed us the power of this technology. It's been around for a long time, but it finally kind of hit the ground running. Regenerative medicine, therapeutics, diagnostics, obviously vaccines, agriculture, and really importantly, the ways of getting the RNA to the right cells at the right time is also really important. And the reason it's become so popular so quickly, A, it's very rapid, it's iterable, it's easy to switch things around, change the code and make new drugs very, very quickly. It's very easy to scale up and it's very cheap. So it really has a lot of promise as a therapeutic platform, not just as a vaccine platform. The way we're doing that, and this is really one of the most fun parts of my job, is I get to sort of help build this thing. We've set up a company called RNA Australia, and under that company, that's funded by government, but it's at arm's length from government. Under that company, we're going to have this manufacturing facility that's being built at Macquarie University. We will have a company called Milo who are going to run that. They're a biotech company from Boston. They're going to run that facility on behalf of us. They will put their own material through. They build cancer immunotherapeutics. But there's reserved capacity in that facility for any research that comes out of Australia that wants to go through, that needs material for clinical trials, can come to that facility with funding and put it through that facility. So it's a really important piece of research infrastructure, if you like. And then alongside that, we've got money to put into an investment program. We're hoping to set up a kind of a venture capital fund, essentially, that will fund research. The whole idea is we'll get new products, new technology. We'll also get, hopefully, some spin-out companies and new intellectual property. But it's a really interesting example for me of how, in the faces of a crisis, science really came together and coalesced around a particular technology. And a bunch of scientists who would normally be competing with each other, and it's pretty cutthroat, like any scientific field, really came together and cooperated and have now built this amazing kind of momentum by working together instead of against each other. I think it's a really important lesson for the scientists out there. And one of the reasons we're very optimistic about this in New South Wales is because we already have a really strong RNA research background in New South Wales, so in the basic research, but also we have an amazing clinical trial system in New South Wales. It's recognised around the world. Companies come here to do clinical trials for lots of reasons. And so we're hoping that these two things can work together. Okay, enough about that. What I want to do now is to talk about Justin. Now, a lot of you in this room know Justin or knew Justin. I love this photo of Justin. It's one of my favourite photos of Justin. Justin and I, like a lot of people you've heard from today already, were mates before we were scientific collaborators. And here's a picture of us mountain biking down in Thredbo a long time before we started working together. But this was really the genesis of us working together. We were very late one night after mountain biking. We got chatting about research. I was working on pancreatic cancer at the time and working on how protein metabolism works in pancreatic cancer. Justin and I got chatting and he's like, oh, we could use some of that to understand motor neuron disease, right? Now I'd known Justin, we'd crossed paths socially for years, but we'd never really talked about putting our heads together in a research capacity. And we very quickly realized there was a lot of synergy and a lot of crossover in what we did. Justin was really good at building tools, at building methods for looking at things, and I was doing that in a cancer context. And so, 15 years into my research career, working on cancer, trying to disrupt proteostasis to kill cells, I had to switch, start thinking about how to maintain proteostasis and keep cells alive. So instead of trying to kill these horrible cancer cells with new drugs, suddenly thinking about how do we keep motor neurons alive, but targeting the same pathways in the cells, the same biochemistry, the same genetics. It was a really interesting shift. And there are a lot of things that I knew from working in cancer that I could bring into motor neuron disease research and vice versa. So there's this really interesting cross-fertilization of ideas and ways of thinking. And for me, it was really eye-opening to see just how embedded certain ideas become in a field where people can get very myopic very quickly. And that was a really important lesson for me to learn. So, you can all read Justin's papers. Rachel went through all the awards and everything he's got. I'm not going to run through that. I want to just talk a little bit about the ideas behind what Justin worked on and what he found and what the impact of that is. Okay, so some of you will know what proteostasis is. It's the maintenance of the proteome. The proteome is all the proteins in a cell. They're the workhorses. They're the machines that do things in cells. Proteostasis is simply the maintenance of the proteome in its right shape, its right amount, and its right location in a cell. It's as simple as that. But that seems really simple, right? But the numbers behind it are absolutely staggering. So you have somewhere between 50 and 70 trillion cells in your body, right? In your central nervous system, your brain and spinal cord, billions, hundreds of billions of cells roughly. A single one of those cells contains billions of protein molecules, right? So the numbers are literally staggering. To do that, you need 3 million ribosomes, which are these RNA machines that make proteins. There's 3 million of those in a single cell while we're sitting here talking, doing that at 6 amino acids per second. Amino acids are the things that you put together, like the Lego bricks that make proteins. All of that takes 3,000 molecules of ATP to make one protein. And what that means is that making protein and keeping protein in the right place at the right time in the right shape uses about 3/4 of our total energy consumption of a cell. It's probably one of the most important things that a cell does. And I like this analogy, and I use this analogy because this was Justin's favorite analogy. When you look up at the night sky, particularly if you're out in the country in a dark sky, when you look up in the sky and you see all those stars, the number of stars we've been able to count in the Milky Way, just in our galaxy, is roughly the same number of proteins there are in a single motor neuron, a single nerve cell. I just find that kind of staggering to wrap my head around, to be perfectly honest, but it's also a nice connection. You know, Rachel talked about nature and connection to nature. It's a nice connection between nature and the inner world, I guess, of science. The other thing that's important to know is that a huge proportion of our genome, a lot of the genes that exist in our genome, their job is basically to keep those proteins folded and in the right place and keep them from damage, or if they get damaged, get rid of them and clear them out. So thousands of different proteins exist to maintain proteostasis. And the reason that's important is because you need a balance in proteostasis, right? Proteins get made, they unfold, they get damaged, they need to be taken care of, and there needs to be a balance in that. But as we age, and in disease, in cancer, motor neuron disease, Alzheimer's, lots of different diseases, diabetes, that system gets out of balance. It loses homeostasis, it loses balance, and it goes out of whack, and that's where the sort of fundamental, or one of the fundamental underlying processes driving motor neuron disease comes from. And that's what Justin and I worked on for a long time. And the reason it's important, we know a lot of the genes that have been identified as being important in ALS and motor neuron disease are involved in those processes of maintaining proteostasis. And we know that one of the pathological hallmarks of the disease is the presence of aggregated proteins, clumped up proteins, a bit like scrambled egg or boiled egg white, right? So it's a fundamental process. And, you know, running with the space analogy, what happens when these proteins tangle and clump and form these inclusions is they're kind of like a black hole. They suck in other proteins around them. And with that, they suck in biochemical information. So as those proteins tangle, they stop working properly. They bring in other proteins, and essentially, like a black hole, which sucks in everything, including light, including the information that's encoded in that light, they're like the black holes in the middle of a cell, these protein clumps. They suck in information. And eventually that overwhelms the ability of the cell to function, and the cell stops. So the sort of questions Jay was interested in are these. Why and how do proteins aggregate? Really fundamental, like really basic, simple questions about what's going on in a cell, what happens normally, and what goes wrong in disease. Why and how do proteins aggregate? Why are some proteins more likely to aggregate than others? How do cells deal with protein aggregates? What happens inside a cell when this happens? It's not just biochemistry, it's cell biology. And then really importantly, can we target these processes for therapeutic intervention? And the point I want to make here, those top few questions are basically curiosity questions, right? They're questions of innate curiosity. How does this work? Without answering those questions, you don't get to the bottom bit, is how do we target these things for therapeutic intervention? You know, and there's probably an argument to be made, and we heard Rick, you know, that amazing talk that Rick gave, and it's a recurring theme in a lot of diseases. When clinical trials fall over, we often come to the conclusion that, well, we probably don't understand the biology or the underlying pathophysiology well enough. You don't get that knowledge without those basic science questions. Now, unfortunately, the way we fund science, not just here but overseas, is moved away from funding those fundamental questions. It's very hard to get money to do that, and I can see nodding heads in the audience. If we don't do that, the pipeline for new therapies dries up. Okay, we need to, and that's why we all need to advocate about this. Discovery research, curiosity research drives new therapeutics. It can't all be translational research. Okay, so the kind of insights that Justin got, and like, to be honest, to try and capture an entire research career in 2 slides is impossible. I'm not even going to try. But you know, what the sort of notions that we were working on and the things that we would often map out on beer coasters at conferences late at night was to try and think about things differently. What are the unifying things of all these different bits of thread that we know about motor neuron disease that all of you work on, you know, whether it's metabolism, whether it's RNA, whether it's protein trafficking, whether it's autophagy, all these different threads. What happens if we try and weave them together? Can we come up with some kind of overarching picture of biology and what's going on? And that led Justin down a really interesting path where he started to think not just, okay, is it the aggregates killing the cells? Not just Justin, lots of people thought about this. Is it not just the aggregates killing the cells? Maybe these aggregates are causing some sort of system failure, right? They're causing a resource deficit. So by sucking in all these other proteins into the black hole, maybe that's stopping all these other things that need to happen in cells from happening. Because the question that we kept coming back to all the time is that this disease manifests often later in life, but the processes, the underlying biochemistry are going right from the start. Got 2 more slides and then I'll stop. They're going right from the start. Why is it that it's not too much later that the disease actually manifests, right? And again, you can read all the detail in the papers, but you know, by understanding this, by thinking about thresholds, when do things get over certain thresholds, then we start to identify bits of, I guess, bits in time where we can start to intervene. And you heard Chris talk about it yesterday, you know, Luke, Jeremy, everyone's thinking about it in this context. All right, so last 2 slides. I sat down the other night and I just wrote down some words that came to mind when I thought about what it was like to work with Justin as a scientist. And I want to reflect on these because I want to try and encourage all of you to think about these things in what makes a good scientist. I love this picture of Jay. This is us at a conference in Croatia. And one of Jay's superpowers was connection, right? And a lot of you would have experienced this. Like a black hole, he had this amazing gravity. He could suck people into his world, into his research questions, and bring in all his expertise from all around the world. You know, I'll say including Nobel laureates and people who will probably be Nobel laureates in the future. He had this amazing ability. And we used to run this amazing tag team show at conferences where we take it in turns to stand next to a poster. We go and grab beers and offer beers to people and say, come and look at our poster. And that I love that picture because it captures it really beautifully. They're all the words up there that describe what it was like working with Justin, right? And many of you will relate to those. It was also incredibly challenging in later years to work with Justin, right? It is impossible to describe what it's like to be trying to cure a disease that's taking your mate away from you before your eyes while you're working on it. And I think impotent is the best word I can think of. But I think all of us realized, including Justin, that we weren't working for him, we were working for who came next. I think that's also an important thing to keep in mind. But he was, you know, he was passionate, he was creative, he was fearless, he was courageous, and he was empathetic. You know, he had a huge heart and he cared about the people that worked around him. And so that's the bit I want to leave with. And I kind of summed it up with the thing at the top there, WWJD. What would Justin do? Right. I often find myself thinking, whether it's in a professional context or a personal context, how would Justin deal with this situation? Because he had an amazing way to think about things, right? And to focus on the bigger picture, to really get to the heart of a matter and understand what was important. And with this slide, I really want to talk to all of Justin's lab and everyone else out there who's working on this disease. To not second guess yourself. The one thing that Justin did as a scientist, he was fearless. He had bold ideas and he was never scared to chase those bold ideas. And I think that's a really important message to take, right? Don't second guess, particularly like, you know, I'm looking at you. Sorry, Jeremy, I'm looking at you. But don't second guess, oh, what would Justin want me to be working on? He would want you to work on the ideas that you've got yourself. Because what he was most proud of was building all of these amazing scientists, junior scientists, supporting them and helping to build their careers. So a little bit of a sort of pointed message, I think. And I'll just leave you with this. And again, this is my second favorite photo of Justin, because this is what we would often just go for a walk and have a chat. And, you know, he had this amazing interaction with Steve Hawking. Steve was an incredible motivation to Justin. And I think this captures that motivation really nicely, right? Look up at the stars, not down at your feet. Try to make sense of what you see and wonder about what makes the universe exist. And that's the essence of being a scientist, right? And be curious. Let that curiosity drive the research. I think that's the message Justin would want us all to hear. Thank you.